Hi there. Welcome to the From Lab to Launch podcast by Qualio, where we share inspiring stories from the people on the front lines of life sciences. Tune in and leave inspired to bring your life-saving products to the world. Hi there. Welcome to the From Lab to Launch podcast by Qualio, where we share inspiring stories from the people on the front lines of life sciences. Tune in and leave inspired to bring your life-saving products to the world.
Welcome to another episode of From Lab to Launch by Qualio, where we delve into the latest innovations and insights from the life science industry. I'm Meg, your host, and today we're thrilled to have Dr. Rolland Carlson, the CEO of Immunexpress, with us. Rolly has been at the forefront of transforming sepsis diagnosis. Sepsis is a critical condition responsible for millions of deaths and significant healthcare costs globally. Rapid and accurate diagnosis is paramount, and Immunexpress has developed a diagnostic tool that significantly enhances the ability to differentiate sepsis from non-infectious inflammation within an hour. Rolly has over 25 years in biotechnology, serving as CEO a few times, and held key roles at Abbott Laboratories and Visus Inc. in molecular diagnostics and business development. You can see his full bio in the show notes. Septicite Rapid, supported by extensive clinical trials and peer-reviewed publications, has received FDA 510(k) clearance, CE marking, and approval from the Australian Therapeutic Goods Administration. Welcome, Rolly.
Matt, great to be here. Thank you.
Thanks. So you've been the CEO of Immunexpress for over 6 years. Can you tell us briefly how you got connected to the company and what it was like leading through COVID? It was probably a disruptive time for the businesses, I imagine.
As it was for everybody, I'm sure.
Yes.
Yeah, I joined the company actually after a career at Abbott, as you mentioned, where I had both by diagnostics and pharmaceutical responsibilities, and I left Abbott to join the entrepreneurial world. So, and I left for startups in Austin, Texas, and we founded a company called Asuragen, which now has been purchased by Biotechne, and later a NASDAQ CEO company for Waverton Biosciences, and now Immunexpress. I knew Immunexpress as in my company in Austin. We had built an assay based upon their novel gene markers for sepsis, and they wanted to have a system by which they could do clinical testing for. So I was introduced to the company that way, and after my stint at Waverton, I was gainfully unemployed, and the board was looking for some changes of leadership. And so they asked me to be an advisor, and one thing led to another. I came on as CEO.
Stars aligned.
Yes.
Yeah. And what kind of challenges did you all face during COVID?
Well, with COVID, I think as we were in the middle of our clinical trials associated with our diagnostic, we developed a product, and we were quite bullish about, from a timing of not only fundraising, but then also getting FDA clearance. And COVID put a wrench in the works quite a bit. Not only did it slow down, obviously hospitals were the front line was COVID management at that point in time, and doing clinical work was not that high in their priority. And so, however, we knew that the test, and we had developed and did studies in Europe very quickly that found that the test was very usable for COVID-induced sepsis, as a result. So I delayed our clinical trials for probably a good year, and then also we filed for the FDA, and of course they were looking at approvals for COVID products, and so that delayed us another 9 months or so. And so, but we were fortunate; we were the first non-COVID product in infectious disease to get cleared by the FDA, coming out the other end, and so we were very pleased with that.
Lots of stars aligning on your side there.
Right.
So Septicite Rapid is a groundbreaking tool in sepsis diagnosis. Can you share with us the inspiration and initial vision behind the development?
Yes, well, sepsis is really a conundrum, where it's one of the leading causes of death in U.S. hospitals. There's been some improvement as far as mortality and morbidity, but it's very difficult to diagnose. And so physicians actually are relying on pretty archaic technologies from 100 years ago of doing blood cultures and waiting for 24 to 48 hours to see if they can get a result, and many times they don't get a result. So, physicians are really challenged to have a multitude of inputs and to try to diagnose sepsis early. It's easy to diagnose sepsis when it leads to organ failure and somebody's in dire straits. However, early detection of sepsis is quite difficult. So therefore, there's a lot of judgment that physicians have as a consequence of that. So the vision of our technology was to actually develop a test that could be rapidly diagnose sepsis in the early stage and be able to intervene with the appropriate patients who are septic. But many times, 42% of the time, physicians believe it's sepsis, but it's not. So both ruling in and ruling out sepsis is very, very important, because it could be these people are sick, there's some other etiology, they should be treated for something else, or they should look for something else. So the way to do that, which historically has been to look for pathogens, requires a number of copy numbers of a bacteria or virus or pathogen or a fungus to grow. And what we were measuring, we found that the human sepsis is really a dysregulated systemic inflammatory response syndrome as a result to a pathogen infection. And what that starts is your body is actually hyper immunoreacting to the pathogen. And it's really that hyperimmune response that actually leads to cascade and ultimately, high morbidity or mortality. And so what it was important to do, there's systemic inflammatory response with no pathogen. It's ephemeral. If you're in a car accident or you have chronic pancreatitis, you might have symptoms of systemic inflammatory response. And what we developed was actually measuring the human response on white blood cells with a highly sensitive mRNA qPCR test that measures differential expression of these genes. And these genes, what they're able to do is, some in the presence of a pathogen are highly upregulated and the other one is not. And then if there's not a pathogen, then the one gene is quite stable and the other one is actually lower expressed. So we can measure the algorithm and that spits out a score of the probability of sepsis from high to low.
Right. And how is the speed important here with this new testing compared to the old archaic methods?
Well, it's pretty well known that if somebody does have sepsis, as each hour goes by, the increased probability of mortality goes up by 8%. So if you're waiting for 12 hours or so, obviously that's a problem. And so it was important when we did our market research is that you needed to have a test. And the test, by the way, that I originally developed in my former company for Immunexpress was on a conventional molecular platform, which was a plate-based one, which would take about 8 to 12 hours. And they were looking at retrospectively what was the diagnosis and found a high correlation. But from a marketability standpoint, you really had to have something that could have a result within 1 to 2 hours. And so that was part of our design goals for the test that we have, Septicite Rapid, which is a sample to answer product and very easy to use, and we can have a turnaround time in 1 hour.
That's a big difference from 8 hours.
Absolutely.
That's such an improvement in patient outcomes, I can imagine.
Indeed.
What else with the Septicite Rapid performance compares to other biomarkers that you can share with us?
Well, there's a number of biomarkers, and there's other sort of symptomology, that in clinical science that physicians look at. And in sepsis, there's probably about 12 of them. It'd be fever, high white blood cell count, low lactate levels. Lactate is something that is used. That's more about perfusion and make sure that you have the appropriate sort of fluids associated with that. And other biomarkers are non-specific. In other words, there's some procalcitonin, which is actually a marker that is very specific for bacterial infection, and is not used. It's used indirectly for sepsis detection, but it's labeled for actually withdrawal of antibiotics if somebody is responding to that. CRP is also used in some occasions. But what they have to do is really take all of those together and be able to come up with a result. And in our clinical trials, what we did was we looked at our Sepsis score result, which is the result that we get from our test, compared to all those other variables. And it was, while when you combine those variables together, then you get an improved performance. The Sepsis score alone was actually superior to all those combined. So instead of a physician having to do multifactorial in their head of those 12 different things, then they should be able to have that score. Now, you do need to look at those other variables because every patient has sort of a unique situation. And so, so what we look at is if there's two sides of that systemic inflammatory response, and physician is suspicious of sepsis, then you should do a blood draw, and which normally they would do for cultures. At the same time, they should do this for our test. And what we found was that within an hour you can have the result with a very high correlation. If it led to a positive blood culture, we had a very high high correlation with blood cultures. But we had that result within an hour, where you had to wait for 24 hours for the blood culture.
And time is of the essence with sepsis. So that's
Absolutely.
amazing. Well, we talked a little bit about your challenges with COVID. What are some of the most significant challenges you faced in the development and clinical validation of Septicite Rapid?
Well, in my career, and I'm sure that your listeners as well, if you're looking at a product that's you're trying to develop, and you're actually looking at something that is maybe a better mousetrap, maybe it's faster, cheaper, whatever, for an existing product. What I'll call the product market fit is pretty well defined. Okay, so do you have a better oncology test, something that's faster, for EGFR or KRAS or something along those lines, or easier to handle? So that is fairly straightforward. But when you get to a product which is in a space that is novel and there isn't a fixed application, then that product market fit is very important to understand early in the process, because you can go down and think, internally believe that, this is the best thing since sliced bread. But as a matter of fact, are you really fulfilling a customer need? So you have to do the market research up front. You need to make sure that your design goals fit that. And you have to be pretty rigid about that. Do you meet those or not? For example, if we were trying to develop Septicite Rapid and it took 3 hours instead of 1 hour, then you know what? You should go back to the drawing board. Don't try to go out with 3-hour tests, in that regard. And then the other thing is do the market research up front, and then and I think that as you're doing in the IVD world in diagnostics, it's different than what you're doing for a CLIA test for sure. You need to be able to lock down what the specifications are for getting FDA clearance and other clearances. And many times companies, because of the rush to try to get the product out, will lock down those design goals too early. And then they get down the process and they might find that manufacturability is a problem, things along those lines. So my philosophy, which I've learned the hard way, is stay in feasibility as long as you can to make sure that you've asked the hard questions that you're able to achieve as far as those goals are concerned. And then actually it's very easy to go through that design process afterwards.
Okay. Great advice. Speaking of your 510(k) clearance and CE marking, you've also gotten the Australian Therapeutics Goods Administration approval. That's quite the regulatory quality achievements, and quality management is at the heart of what we do here at Qualio. So have to ask, how does Immunexpress ensure the reliability and accuracy of your products and instill a quality of culture?
Well, I think that number one, if you're in this business, you have to aspire to have the highest quality to fulfill your customers' needs. And we're talking about serious, medical devices and diagnoses, and you have to ensure that you're providing your customers and your patients, quite frankly, the highest quality product. I had the benefit of learning within the Abbott system, so to speak, that you needed to really be in lockstep with quality and regulatory early on in the process. And particularly for founders, and I've worked with founders who had for example, the company in Austin that I joined, the co-founder there was, a fantastic scientist and very successful in the life science world, but had never done anything in the quality or regulatory sort of, and the appreciation of that, one tends to discount if you haven't gone through that, but that can come back clearly to be an issue. So engaging quality and regulatory, which I've, as key members of the team, not just something that you sort of throw over the wall. You can't do that, to be successful, in that regard. And I've been very proud of the team that we have, in achieving the accomplishments we have at Immunexpress. They're experienced both domestically and internationally. And, if you work with the agencies up front too, and making sure that you, in some cases, and particularly in this situation where you have a novel product like Septicite Rapid, you need to go through as engage the agency early on and saying, this is what we're planning on doing. Do you have the input before we spend, millions of dollars on clinical trials, et cetera, like that.
Yeah, great advice. Those early FDA meetings are always worth having, especially with those novel devices, to know where you need to chart your map.
Indeed. Lovely. And sometimes you get feedback that you really don't want to hear, but it's very important to get that early on, because if you spend, your investors' money and then you have expectations for clearances and you have a hiccup, and I think there's some experiences, fairly recently with companies where they went all the way down the path and they couldn't get FDA clearance, and that obviously is very
too late to pivot at that point. So
Exactly.
How does Septicite Rapid test integrate with current clinical workflows in hospitals, and what feedback have you received thus far from healthcare providers?
Well, what we found with sepsis, and not only in Europe and in the US, it's managed very differently by different institutions. But there's many sort of stakeholders associated with sepsis management. If you're trying to be able to identify sepsis in the emergency room department, then what you're trying to do is, quickly rule in, rule out in that regard. Our test is really not for screening, a patient who might come in with a fever and looks a little sick or so. It's really somebody that's strongly suspected of being sepsis. And the different stakeholders associated with sepsis management are going to be critical care doctors, nurses, ED doc that's going to be doing the admissions, and then ultimately, the infectious disease doctor that after patient is admitted. And what happens is initially there's going to be an order set. Different hospitals will say, if I suspect that there's going to be a patient with sepsis, they'll say, I want to draw blood for lactates. We're going to look at WBCs, white blood cell counts, and they call these things sepsis bundles. And there are sort of 3 or 4 key criteria by which they're going to react to. And there are international guidelines associated with that, but they're they are not something that everybody follows. In the U.S., CMS has actually been pretty prescriptive as far as what they call SEP1 bundles, where if you suspect somebody to have sepsis, you need to either administer antibiotics and treat them within 3 hours or not. And you need to know, 1, is there an infection, and 2, is there a systemic inflammatory response, which is exactly what our test does. So what we're doing is when we approach customers, what are you doing for sepsis? 2, what is your need? Because in some cases, it's the inpatient sepsis that's their biggest problem. They've got their process for admitting, et cetera, like that but when a patient ends up in the hospital, then what happens to them? And in other cases, we have too many people coming through the ED. We can't triage them fast enough, so we want to be able to bucket them. That's associated with it. So we try to, and many hospitals want to do an evaluation. I mean, for us, it's important to do an evaluation to try to meet their unmet need, right?
Mhm.
So that's our approach. I think that our test really helps drive compliance to the CMS step 1 sepsis bundles, and I think there's a good recognition from hospitals associated with that. And I think that as we have used the test is being used, there's some interesting results. First off, I made that comment about 42% of the time physicians think it's sepsis and it's not. So that's being verified. And so major customers are saying, wow, we're really surprised how many people we thought had sepsis don't, but then on the other hand, they're saying, wow, you guys really highly correlate with blood culture if it's positive, and we're ruling in. Another use case would be post-surgery. If you have patients that are having surgery, they'll be in a surgical ICU. There's a lot of concern about infection, and can they be really discharged to the ward for recovery? And looking at our test as far as rule in, rule out is important. And if our test is positive, and I'm not sure if your listeners are aware, but right now there's a blood culture bottle shortage right now that the FDA has put out notice for. And the blood culture industry is still very, very large. But right now there's a shortage of that. And what we found is that with our test for positive, actually you would like to double up your blood cultures to look for that causative organism, whether it's that there might go down sepsis. You start treatment, but then you want to find the causative agent in that regard. But on the other hand, if we have a low probability of testing, then perhaps you shouldn't be having to administer all those blood cultures, and you should be looking for something else.
Back to that 42% that you mentioned of doctors thinking it's sepsis when it's not. How does that play into being good stewards for antibiotic stewardship and fighting against antibiotic resistance?
A very good question. And it's one where, the other thing, CMS is actually looking for compliance to, antibiotic utilization, because I think as your listeners probably are aware, basically resistant organisms within a hospital is a big problem. And quite frankly, it's anticipated the number of deaths associated with antibiotic resistant organisms is going to exceed those of cancer deaths by 2050. So this is something that very much needs to be addressed. But I understand the situation. Physicians are certainly in that they don't want to. They don't want to miss a patient. If they believe it's sepsis, their only recourse, I'm not really sure, but my default is I'm just going to be able to put them on antibiotics. And I think that, and frankly, it's up to us in the biotech world to come up with solutions to help them, give them the tools to provide this selection criteria. And these are busy people. They're very, very smart. They know what they're doing, but they have. If they have limited tools, then they're going to stay with what they do.
Mm-hmm. Yeah, to stay that conservative approach and treats. It's amazing work that you all are doing. Looking ahead, what are your future plans for future developments or new innovations in the field of sepsis diagnostics or beyond?
Well, I think with sepsis, faster, cheaper is always useful. Our next generation product is Septicite Rapid BD bacterial viral. So in a single cartridge, of which we, basically, in my hand, I'm holding a molecular lab in my hand here. And this has got all the reagents, everything that's necessary for extracting, purifying the genes, and then amplifying them, and then being able to have a report out. And to that, for the question that happens is, okay, if sepsis, and our test, by the way, says sepsis, it could be based on viral, as I said, with COVID, bacterial, or fungal. And we just have found for parasitology as well. But the main drivers are bacteria and viral. And so our test, that we're combining genes, host response genes, that basically are saying if it's sepsis, then is it bacterial or viral? And you'll have that same result at the same time. So that's what we're doing. We're expanding also into pediatric indications. And I think, combining this with, direct detection of pathogens in the future will be very, very powerful.
I can't wait to see what you all do next. Well, Rolly, our last question is more of a fun one we like to ask each of our guests. If we ran into you at the bookstore or at a local library, in what section would we find you?
Well, my PhD, I'm actually a plant biochemist that focused on marine biotoxins. And so I love to dive. I love the ocean. I love anything above or below the ocean. So you'll find me either in the sort of oceanography, certainly the marine environment, et cetera, along those lines. So, and quite frankly, I love geography. I love history. But my passion, outside of what I do for work, is certainly water oriented.
I'm a former ocean girl myself, so I love the ocean too. Yeah. Well, it was great to have you on the show today. Where can people go to learn more and follow along and connect with you, Rolly?
Well, you could look up our product at septicite.com. And certainly the company is Immunexpress.com.
Terrific. Thanks so much, Rolly. It was a pleasure.
Enjoyed, Meg. Take care.
Thank you.
Thank you for listening to this week's episode of From Lab to Launch, brought to you by Qualio. If you like what you've heard, please subscribe and give the show a positive review. It really helps us out. For more information about Qualio, our guest today, or to be a guest on a future episode, please refer to the show notes. Until next time. Thank you for listening to this week's episode of From Lab to Launch, brought to you by Qualio. If you like what you've heard, please subscribe and give the show a positive review. It really helps us out. For more information about Qualio, our guest today, or to be a guest on a future episode, please refer to the show notes. Until next time.